And our final talk is phase shift photoacoustic spectroscopy, light absorbing aerosols by Gwen Lawson of the University.
Yeah, thanks. So hi everyone, I'm Gwen and I'm going to talk to you a bit about my project looking at the optical properties of light absorbing aerosols here in Bristol. So just to start with a bit of background, aerosols in the atmosphere can interact with sunlight both directly through absorption of light or scattering of light, and there are also indirect effects such as altering cloud albedo. So we can quantify aerosol optical properties using their cross sections, given the symbol sigma. So the extinction cross-section is the total amount of light removed by the aerosol particles, and that's the sum of the scattering and the absorption cross-sections, which is the amount of light removed by scattering and absorption processes. So we can calculate these optical cross-sections from the complex refractive index of a particle, given the symbol n. So that's a sum of the real component n and the imaginary component k. So we use something called mean theory to calculate these optical cross-sections. And then inversely, if you measure these optical cross-sections, you can then retrieve a complex refractive index value by basically doing a grid search over a range of expected values to see which one best fits your data. So the sort of motivations of the project are the fact that we don't very well understand the role of light absorption when it comes to radiative forcing. So this is a figure from the IPCC 2021 report. We can see, particularly for black carbon, which is strongly light absorbing, the error bars here are quite big and they even span both the positive and the negative sides of radiative forcing. So we really need to improve our understanding more to help improve climate models and to understand how we affect radiative forcing. So the aims of my project were initially to look at dry aerosol particles, to measure the extinction and the absorption cross-sections of these. And I looked at ammonium sulfate and nitrogen and also mixtures of the two of these. And I used an AAC for size selection by aerodynamic diameter. So nitrogen is a really strongly light absorbing organic dye. It looks basically black. And then if we mix that with our non-absorbing species, our inorganic ammonium sulfate, we can basically produce a range of different absorption strengths from purely non-absorbing ammonium sulfate to really strongly light absorbing, and we've just got nitrogen. So from these measures of extinction and absorption cross-section, we can retrieve refractive index values across the mixing range, and then use this to look at some different mixing rules that are usually used to predict refractive indexes and see how well they fit our data. And then we want to look to extending our measurements for looking at hydrated aerosols, as this would be a lot more atmospherically relevant. So this is a schematic of, I think it's died. It did have no battery. Yeah, so this is a schematic of our experimental setup. So we make up our stock solution of our desired composition, water, we atomize this, we've then got a pressure buffer to help maintain downstream pressure, we then dry our aerosols, and then we size select them using the AAC, which is the aerodynamic aerosol classifier. And we do this between 100 and 400 nanometers in 25 nanometer increments. Then we measure our mobility size distribution using an SMPS, scanning mobility particle sizer. So if we've got measurements of our aerodynamic diameter and our mobility diameter, we can then calculate the density of our particles. So once we've measured the size distribution, we then go on to measuring the optical properties. So these are our spectrometers. They're all contained within an instrument that was developed by the Met Office for field and lab experiments. So we've got two different sampling lines at different wavelengths of laser light, 658 and 405 nanometers. So the aerosol sample first passes through the cavity ring down spectroscoper, which measures the extinction, and then it goes into the photoacoustic spectrometer for absorption measurements. And so I mentioned that we want to get these values of cross sections. These are single particle measurements, but because we've got an ensemble aerosol sample, we also need to know our number concentration, N, because these measure the optical coefficients when you've got an ensemble present. So we also have a condensation particle counter, CPC, on the back end of our photoacoustic spectrometer to get our number concentrations, so we can convert our coefficients to our cross sections. So a little bit more about the techniques. This is cavity ring down spectroscopy. So it's a really highly accurate technique, which is calibration free. Basically got two highly refractive mirrors either end of the cavity, so you can bounce the laser light back and forth between the two. A small amount of light leaks out the mirrors, and we can measure this using a photodiode, and the characteristic time scale of this decay is measured. We first of all do this with no aerosol present in the cavity. That gives us tau naught. And then we do the same thing with our aerosol present. And because you've got aerosols that are going to absorb light, you then have a lower timescale of decay, tau. So then we can use this equation here if we know a few things like the length of the cavity, which we do, and also the length occupied by the aerosol sample, because we don't want the aerosol occupying the full length of the cavity because you start to get aerosol depositing on the mirrors, which affects your accuracy. So if we know those two things and the speed of light and our two tau values here, we can then get our extinction coefficient. So then photoacoustic spectroscopy is what we use for our absorption measurements. So we have a periodically modulated laser that we inject into our cavity. It's got highly refractive mirrors at the end again. So if you've got light absorbing aerosols present in the sample, they'll be heated by this laser. They then transfer their energy to surrounding bath gas, which causes the bath gas to undergo adiabatic expansion. And because we've modulated this laser periodically, you get periods of expansion and contraction, and that basically generates a sound wave. So we can amplify that and detect that using a sensitive microphone, and then we can relate this amplitude of our microphone response, which is this red trace here. We can relate that to our absorption if we calibrate our spectrometers with something with a known absorption. So we do that using ozone. So here are some results for the refractive indexes we get using our measurements of extinction and absorption cross-section for our pure aerosol components. So on the left here is the real refractive index N for ammonium sulfate. The points in blue are my values, and then we've got some comparisons made with these red values, which are using the same spectrometers, but instead of using an AAC, they used a DMA for size selection, which introduces multiply charged particles. So we were hoping to improve our precision, which we do when we take into account, so we've only got one CPC, so we've located ours on the 405 nanometer channel, but previous work used the 658 nanometer channel. So the precision of our refractive indices depends quite strongly on which channel you have the CPC on. So in an ideal world, you'd have two, but only got one, so that affects the precision. And then we've got on the right here, nibrazine, which is our really strongly absorbing compound. At the top is the real refractive index. Again, blue is this work, red is the previous work using the DMA for size selection, and then the black trace is using a slightly different technique to measure absorption, but they've done it over a wide range of wavelengths. And then again, in the bottom plot is the imaginary refractive index K. So we see pretty good agreement with the literature values. This here sort of deviates a bit, but one thing to note about nibrazine is that it's not a specific mixture of compounds. It's basically formed as a byproduct of a reaction, which is then color matched. So the actual structures vary from batch to batch and between manufacturers. So we would expect there to be different optical properties between those. So then when we go on to looking at mixtures of the two, we've got along the x-axis is the mass fraction of nibrazine. So we go all the way from 0, which is our pure ammonium sulfate, to 1, which is our pure nibrazine. And these points here are our experimental retrieved N and K, real and imaginary refractive indexes for our two different wavelengths of light, the 658 in the red and the 405 in the blue. And then, so quite often in the literature and in climate models, they use linear weighted mixing rules to try and predict the refractive indices of these binary mixtures. So here I've shown the mass fraction mixing rule, which basically just takes a mass fraction weighting of the pure component refractive indices. And we can see that particularly for this N, we see a highly non-linear relationship here between mass fraction and N. And only when you use a different kind of mixing rule, which is called the molar refraction mixing rule, which takes into account the density of the particles, because we're mixing an inorganic and an organic, you don't get nice ideal mixing, you get deviations away from this. So when we input our densities, which we're measuring from our AAC and SMPS measurements, we get much better agreement using this molar refraction mixing rule with our results. So that's showing us that you really can't just use these linear weighting rules when you've got inorganic and organic mixtures. You need to use a physically based mixing rule, such as the molar refraction mixing rule, and take into account the density. So one of the problems with photoacoustic spectroscopy is that at the moment it only provides accurate measurements for dry particles that haven't got any water or any volatile organics present. And that's because usually the energy absorbed from the laser by the particle is dissipated through heat flux to the surrounding gas phase, which then results in our sound wave that we measure. But when you've got water or volatile components present, you have this competing pathway, the mass flux pathway of evaporation of water. And this pathway has a lower conversion efficiency. So that basically means that the absorption that you measure when you've got volatiles present is lower than it actually should be because of this competing pathway. So this is fine for sort of lab measurements, but really atmospheric aerosols are going to contain water and other volatile components. And so usually when they take this instrument up and measure aerosols, it's all dried before you measure the absorption. So you're not really measuring the aerosols, how they are in the atmosphere. So if we could extend this technique to be able to use hydrated conditions and look at more atmospherically relevant conditions, then that would be really good. So the next steps of my project are looking at how we might be able to do this. So when you have these volatiles present, you introduce a lag in the, so the red here is the microphone waveform and the black is our modulated laser. And you basically get a lag in the response because of this competing pathway, which is called the phase shift. So we're hoping to be able to use this phase shift to correct our measured absorptions that are low biased, to correct these for a value that's more accurate, which would allow us to make measurements at elevated relative humidity, for example. So there's a few tricky things we need to consider about being able to sort of maintain high relative humidity, avoiding any water depositing on the mirrors, and the microphone response is usually humidity dependent. So there's a few tricky things that we need to get right before we can do this, but this is going to be the sort of next steps of my project. So just to summarize, we've validated our experimental approach for looking at dry aerosol particles using the AAC to select based on the aerodynamic diameter for ammonium sulfate and nitrogen. We've seen good agreement with the literature values, and then when we've looked at mixing rules, we've seen that we need to use a physically based mixing rule, such as the molar refraction mixing rule, when predicting these complex refractive indices, specifically when you've got inorganic and organic mixtures because of the non-ideal interactions. We've identified the importance of extending photoacoustic spectroscopy to look at humidified aerosols, and looked at potentially using this phase shift that's generated to be able to correct for our measured absorption. So I want to say a quick thanks to the CDT and the Met Office for the loan of their equipment and my research group, BART, and I'm happy to take any questions.
Hi, what made you decide on the wafer you did use for testing?
So those are the wavelengths that the Met Office built the instrument at. So that's kind of the decision there.
Oh, this one, okay.
Can I just make sure I understand this molar mixing rule? Because if I understand it, the model that the Met Office uses is a volume mixing rule, not a mass mixing rule.
Yeah.
So how does yours differ from the model that the Met Office uses, and should they be using your?
So the volume fraction mixing rule gives quite similar values to the mass fraction mixing rule that I'd shown. So it's actually something I'm going to be looking at in my placement, because they use the volume fraction mixing rule because it obviously gets very computationally expensive when you've got aerosols that aren't just two component mixtures. So we're going to be trying to use AI to incorporate the molar refraction mixing rule into their model, because they currently use the volume fraction, which isn't great, as we've seen.
Okay, so thank you very much, everybody.